I want to start by saying thank you, and today I'm going to be talking about my ongoing work in the Doe lab. Neurons are an essential component of the nervous system and drive animal behavior. In Figure 1a, we can see these Drosophila motor neurons at an early state in development. Motor neurons reside within the central nervous system of the fly, shown on the right, and are important for facilitating a number of crawling behaviors. One of the main sources of support for these motor neurons are glial cell populations, specifically astrocytes. Astrocytes extend fine projections to interact with the connections between neurons that allow them to communicate, called synapses. Astrocytes support synapse function by providing metabolic support through mitochondria in these perisynaptic processes, which are located around the synapse. But what happens if astrocytes are no longer able to provide metabolic support to motor neurons? Amyotrophic lateral sclerosis, otherwise known as ALS, is a neurodegenerative disease that causes selective degeneration of motor neurons, which impacts locomotor behavior, causing paralysis and eventually death. I'm interested in the basic mechanisms which result in an improperly developed nervous system, causing an ALS phenotype in adulthood. ALS is a complicated disease caused by mutations in several genes. I'm focusing on one of these genes called TDP43 in humans and tbph in Drosophila. tbph is an RNA and DNA associated protein which broadly regulates gene expression. In 90% of ALS patients, we see a change in TDP43 localization from the nucleus into the cytoplasm, which you can see illustrated here in Figure 3b and c. Today, I'm going to focus on loss-of-function forms of tbph and its impact on glial cells. As I mentioned, astrocytes metabolically support motor neurons in the healthy nervous system, and we wanted to test the hypothesis that a loss of tbph from astrocytes results in defective mitochondrial trafficking to the perisynaptic processes, resulting in insufficient energy transfer to motor neurons and an ALS-like phenotype. The first step we took was to look at how astrocyte mitochondria are trafficked in response to motor neuron activity. To do this, we used optogenetics, which is a light-inducible way to control neuronal activity. We found that when we activate motor neurons using optogenetics, there's a change in mitochondrial distribution. We specifically looked at synapses in close proximity to mitochondria, and you can see in the quantification here that there is a significant increase in motor neuron synapses contacting astrocyte mitochondria following activation. To test whether tbph is necessary for this activity-dependent trafficking, we performed the same assay in animals where tbph had been knocked down specifically in astrocytes. This resulted in a significant increase in astrocyte mitochondrial volumes near motor neurons compared to control and standard conditions and following optogenetic activation. Because activation of motor neurons results in mitochondrial positioning near synapses, this suggests that knockdown of tbph in astrocytes leads to ectopic motor neuron activity. Though not shown, our preliminary data visualizing motor neuron activity in vivo supports this hypothesis. Thus far, these results suggest that instead of tbph impairing astrocyte mitochondrial trafficking, loss of tbph results in hyperactivity in motor neurons that leads to more astrocyte mitochondria towards synapses. This led us to the question whether this repositioning might be necessary to protect motor neurons from excitotoxicity. In order to test this, we needed a way to disrupt astrocyte mitochondrial trafficking. We know that in general, mitochondria can be trafficked along microtubules using motor proteins, and by performing a genetic screen to remove these motor proteins one by one from astrocytes, we found that knockdown of Milton results in a failure of mitochondria to traffic. What you see on the right is larval brains stained for astrocyte mitochondria in green, and in the control, mitochondria are evenly distributed through the nervous system, but upon knockdown of Milton, we see a failure of mitochondria to traffic to motor neuron dendrites. To understand how this failure of trafficking impacts motor neuron health, we first looked at where motor neurons connect to muscles and found that when we knock down tbph or Milton, there is a disorganization at the neuromuscular junction, and there is also a significant increase in synapse number following knockdown of tbph or Milton. We then looked at how this impacted the behavior of the animals, and by using a high throughput locomotor assay, we were able to see that in the knockdown mutants, there is a significant decrease in overall area covered and speed of the animals, which you can see on this quantification to the right. Now that we have discovered the machinery through which mitochondria are trafficked, we're poised to test whether, in the case of astrocyte knockdown of tbph and Milton together, failed mitochondrial trafficking results in loss of motor neuron populations, leading to an ALS phenotype. I want to thank the Doe Lab, specifically my mentor Sarah Ackerman and Dr. Doe, and additionally I would like to thank the National ALS Foundation and the NIH for providing funding for this project.